Good morning, everyone. Welcome to our series on respiratory pathogens. This is our third webinar on respiratory pathogens, and today we'll be covering Coronaviridae and the Virus Pathogen Resource website. My name is Anna Maria Niewiadomska, and I'm a bioinformatics analyst and the outreach coordinator at IRD and ViPR. And joining us today is Yun Zhang, bioinformatics analyst at JCVI, who will be helping me curate questions, and Richard Sherman, the director of informatics at JCVI, who will also be helping me field questions. In about an hour, Duncan McCannell, our guest lecturer, will be joining us. He is currently at the CDC heading the US sequencing efforts for Sars Coronavirus. And you'll be hearing more from him soon. So I'm just gonna give you a quick introduction to the talk and who we are as we wait for a few more people to join us. Just to get you started, I'm going to give you a quick introduction to the Coronaviridae because while I'm sure most of you are already familiar with SARS-CoV-2 to some extent, you might not be as familiar with some of the other coronavirus species. We'll then move on to a live demonstration and tour of the ViPR website and a couple of the tools that we have for SARS-CoV-2 analysis and other coronavirus-related bioinformatic analysis. Leave some time for questions before moving on to our guest lecture from Dr. McCannell. We'll have a little more time for questions, and I will be available online till about 10 a.m. Pacific time to answer any other questions you might have. We will be recording this session, and we will make it available on YouTube shortly after. So just to give you an idea of who we are and who the bioinformatics resource centers are, they were originally established by the National Institute of Allergy and Infectious Diseases in 2004 with the goal of providing free and public access to computational platforms and tools that would allow users to perform a variety of analyses on genomics data focused mainly on category A through C infectious diseases, as well as host interactions. So as part of our mandate, we also perform outreach, including bioinformatics training at BRC sites and conferences, or for those of you who are interested or your institutions are interested, we also provide customized training based on requests. And we also try to respond to emerging pandemic threats. So for example, we've prepared SARS-CoV-2 portal page, as well as several tools to analyze SARS-CoV-2 data and information. When the BRCs first started, there were multiple different sites focused on different pathogens, but right now they have been joined into two bioinformatics resource centers, the BVBRC, of which we're part of, and that includes the Influenza Research Database and the Virus Pathogen Resource, which obviously are focused mainly on viruses, and our partners at Patrick, who focus on bacterial pathogens. The other BRC, which is called VUPathDB, mainly deals with parasitic and fungal diseases, as well as VectorDB, which hosts data on vectors. And I think we have a couple upcoming lectures, or one upcoming lecture on Aspergillus as a respiratory pathogen from FungiDB, and two upcoming talks from Patrick, as well as two more from us. So I'll get started with a quick introduction to the Coronaviridae family, as a refresher for anyone who might not be familiar. The Coronaviridae family belong to the Riboviria realm, so RNA viruses, and they fall under the order of the Nidoviralis. They're enveloped viruses, and depending on which subfamily they belong to, they can either be spherical or bacilliform. They're about 120 to 160 nanometers across, and their genome is composed of a positive sense single-stranded RNA And for RNA viruses, they're quite large, ranging between 26 to 32 kilobase pairs. So while there's quite a few families and subfamilies and genera in the Nidoviralis order, I'll mainly be focusing today only on the alpha coronavirus and beta coronavirus genera because those are the most relevant to humans. So as I mentioned, the Coronaviridae family is divided into two subfamilies, the Coronaviridae and the Torovirinae. The Torovirinae can infect humans, however, they're not very pathogenic, so they're not discussed often. The genera that do not infect humans have been grayed out, and as you can see, they can infect a wide variety of mammals. But again, the ones we will focus on are the alpha coronavirus and beta coronavirus genera because those are the most relevant to us as humans. So this is a text-heavy slide. I don't expect you to take in all of this. On the left, just put a list of the alpha coronavirus genera. And in this column, you can see a list of representative species. And the ones I've highlighted in orange and red are the only species that, or the only known species that can infect humans. So the ones in orange are considered low pathogenicity. On the alpha coronavirus side, we have 229E, NL63. And on the beta coronavirus side, we have OC43 and HKU1. So these low pathogenicity coronaviruses have a global distribution and are considered endemic and part of the species of viruses that contribute to the common cold, or what's called the common cold, every winter season. On the other hand, those in red are considered to be highly pathogenic viruses. And of course, you're familiar by now with Sars Coronavirus and SARS-CoV-2, which belong to the Sarbecovirus lineage in the beta coronavirus genus. And of course, a few years ago, the Middle East respiratory syndrome, MERS coronavirus emerged, and this belongs to the Mirbecovirus subgenus in the beta coronavirus genus. The primary reservoirs for coronaviruses are considered to be bats and rodents, particularly for low pathogenic coronaviruses. 229E and NL63 are thought to have bat reservoirs, whereas HKU1 and OC43 are thought to have rodent reservoirs. They may or may not have intermediate hosts. Some are unknown and some are suspected before passing to humans. And highly pathogenic coronaviruses, which include SARS-CoV-1, CoV-2, and MERS coronavirus, are thought to have their primary reservoirs in bats. Intermediate hosts for SARS-1 was identified as palm civets and raccoon dogs, and the intermediate host for MERS coronavirus was determined to be camels before they were passed on to humans. The intermediate host for SARS-CoV-2 has not been identified or confirmed. However, pangolins have been proposed as an intermediate host. So while those intermediate hosts have been proposed, I should note that SARS-CoV-2 really does have a broad host potential, and this has been shown in a couple of different papers, and I just wanted to highlight this one because it did a really thorough job of performing an in silico comparison, comparing ACE2 orthologs in multiple different types of mammals, showing that quite a few of them are capable of hosting SARS-CoV-2 replication. And they also did some in vitro analysis. So, for example, if you look here, the SARS-CoV-2 receptor ACE2 is colored in green, and the red color denotes infection of the virus, which is coloring the N protein of SARS-CoV-2. So if you take a look at the ones that have where both are expressed, you can see that it's really quite a broad range of animals. And if you'd like to read more about this, this was published recently in PNAS. So moving on to the Coronaviridae variant structure. They basically have a pretty similar structure, regardless of the virus species. So I'll give a couple of examples and just highlight one specific difference that I think is important to keep in mind. While most of them have similar structural proteins, which include the spike trimer on the outside, as well as two other membrane proteins, the M protein and the small membrane protein, the E envelope protein, as well as the internal nucleoprotein, which protects the RNA genome. There are a few coronaviruses that belong to the Embectovirus subgenus of beta coronaviruses that have an additional envelope protein, and this is called hemagglutinin esterase, or HE for short, and it forms a dimer on the outer surface of the virion and helps the virus attach and enter cells. So this image shows an alignment of the 7 human coronavirus species that we're most interested in. The top 2 belong to the alpha coronavirus genus, and these include 229E and NL63. And we also have the 5 other beta coronavirus species, which include the low pathogenicity HKU1 and OC43, the MERS coronavirus, and the 2 Sars Coronavirus. So as you can see in green, the ORF1 and ORF1b open reading frames take up the majority of the genome, which is about 2/3 of the genome. And then the structural proteins are conserved between all of these different viruses. So the surface, envelope, membrane, and nucleocapsid protein are all conserved between these viruses. But then when you look at other proteins, such as accessory proteins, for example, OR5 or 4, these come in different sizes and positions in the genome and can vary widely even within the same subgenus of viruses. So for example, SARS coronavirus and SARS-CoV-2 have a different set of accessory proteins. So just something to keep in mind. So viral gene expression of coronaviruses occurs mostly in the same way for the Coronavirinae family or subfamily. The first protein that's expressed after entry is ORF1A and ORF1AB polyproteins. Because the viral genome serves as messenger RNA, this is directly transcribed upon entry. And while there's a stop codon at the end of the ORF1A open reading frame, there's also an RNA structure called a pseudoknot that provides a slippery sequence and causes ribosomal frame shift that allows a read-through of the next open reading frame, ORF1b, so that you have extra long polyprotein going all the way from the 5' end up to just about 21 kilobase pairs into the genome. So these two proteins, polyprotein 1A and polyprotein 1A, are produced, and they contain their own proteases, 2 proteases, a papain-like protease and a 3CL-like protease. And these can cleave the polyprotein into multiple non-structural proteins. And these are named NS1 through NS15 and 16. Some coronaviruses have 15 polyproteins, some have 16. The ones we're interested in have 16. And these have multiple different functions, some of which come together to make a replicase and transcriptase complex, which then goes on to transcribe multiple subgenomic RNAs that code for the structural proteins and accessory proteins. And this figure shows the subgenomic RNAs thought to be encoded by SARS-CoV-2. So I will note that a couple interesting proteins that you should know about are RDRP, which is an RNA-dependent RNA polymerase. So a virion encodes its own polymerase, and it also encodes NSP14, which is a 3' to 5' exoribonuclease, which provides proofreading activity that the viral polymerase lacks. So this is one of the reasons why we don't see a lot of variation in the SARS-CoV-2 genomes, or at least didn't see much variation up until recently. A couple of other things that I want to just note are that the Coronaviridae accessory proteins are extremely diverse in their sequence and function. So these proteins, these open reading frames in gray are common between all of these different coronavirus species. But if you look at the colored open reading frames here, you'll see that the position, the size can often be very different. And again, I want to note that they're named in order of where the open reading frame occurs. So if it's just the third open reading frame or fourth open reading frame, it'll be named OR4. But these aren't necessarily the same proteins. It's just where they occur in the genome. So it's worth keeping that in mind and looking at the sequence to make sure that you're looking at the same proteins. On the right, I've provided a list of SARS-CoV-2 known accessory proteins. ORF3A, ORF6, ORF7A, 7B, and ORF8 are well established and have homologs in the original SARS coronavirus. There are a few other open reading frames that have been proposed, but their function is unknown, and it's not known whether they actually are expressed or not. There's conflicting data. So particularly for the ones that have a red asterisk at the end, there is not any strong evidence of their expression in these viruses. So just a quick note on how coronaviruses diversify. Even though they have proofreading activity, they are prone to recombination, and recombination is known to occur at high frequency during replication. You can have intraspecies or interspecies recombination. Intraspecies recombination, so between coronaviruses of the same species, is common, whereas interspecies recombination, while it does occur, is thought to be infrequent. And the S genome is particularly susceptible to this. And as you can imagine, this kind of, especially interspecies recombination, could enable cross-species jumps of coronaviruses. And if you're interested in reading more about this, there's a couple of interesting papers I've referenced here. And again, this is recorded, so you'll be able to slow down and take a look at them if you like. Another method of coronavirus diversity generation is through gene capture. This is rare, but there is evidence of it happening. So for example, the hemagglutinin esterase protein that I mentioned has been proposed to have been captured either from an influenza C virus or a host lectin protein. And again, I have some references provided here if you'd like to read more about them. And another interesting example is in this bat coronavirus, which contains what's called a P10 protein, and is thought to have originated through heterologous recombination with bat orthoreovirus. If you're interested in what the current therapeutics are for SARS-CoV-2, I would direct you to go to the NIH's coronavirus or COVID-19 treatment guidelines page. There's a couple of different antivirals that have been tested, and they either recommend them, detail whether there's insufficient data, or just don't recommend them. And there are also some antibody-based products, immunomodulators such as dexamethasone, and a couple of other experimental therapies that have been tested. There are many and varied vaccines and vaccine types. The first of which I think was inactivated vaccines, which were developed in China, followed quickly by the mRNA vaccines developed by Moderna and Pfizer. We also have adenovirus vaccines, of which there are many examples. The most recent vaccine results came from Novavax, which is a protein subunit, and there are also a couple of DNA plasmid vaccines, which are still undergoing clinical trials. There's also been a lot of recent discussion on the SARS-CoV-2 variants. I won't cover this right now, but we do have a new site that tracks the variant lineages, and these have relevance to the virus's transmissibility and their ability to escape antibodies, and we'll be discussing this later in the talk. So I will end on this slide, which just summarizes the basic characteristics of the 7 human coronaviruses, and check for any questions. Okay, so if you would like to go to the Virus Pathogen Resource, you can find it by navigating to viprbrc.org. This will take you to our homepage, where you can see that we provide data on multiple different virus families, one of which is the Coronaviridae family. If you're interested in researching multiple coronaviruses, you can go there. If you are exclusively interested in SARS-CoV-2, then I would direct you to go to our SARS-CoV-2 portal, which has a couple of extra tools developed just for SARS-CoV-2 analysis for now. You can also navigate through the data a little bit faster because the only genomes that are contained and the only data on this site is exclusively for SARS-CoV-2 species. So we like to think of our sites as separated into 3 different areas. The search for data, where you can look for multiple different types of bioinformatic data, including the genome and protein data for these viruses, which is our most popular. But I'd also like to highlight that we do have some immunoepitopes for SARS-CoV-2 that have either been experimentally determined or have been predicted through using the NetCTL algorithm. We also import 3D structural files that you can view for SARS-CoV-2 proteins from the protein data bank. So let's say you're interested in looking at the structure of the spike protein, you can go in here and find that. We also have a section for analysis and visualization tools, and we also have a workbench where you can not only store your data sets and the results of your bioinformatics analyses, but also upload your own data, merge it with the ViPR data, and just analyze it using the tools on our website. And this is free. There's no size limits. All you have to do is just create an account, and you can go ahead and start working on your data. So I'm just going to give you a quick overview of what it's like to search for data on the ViPR site. You can search by multiple different fields. If you're interested in the DNA genome, you just keep genome clicked. If you're interested in protein data, you would select protein, or if you have a specific strain in mind, you can search using that. You can specify collection year, although for SARS-CoV-2, that's only 2020 and 2021. You can search by geographic grouping, by continent. Let's say you're interested in just North American sequences. You can further drill down and choose which country you're interested in as well. So let's say I'm interested in the early sequences that came out from China and wanted to also look at the sequences that came out in Italy shortly after. You can also select for which hosts you're interested in looking at, although the majority of SARS-CoV-2 hosts are obviously going to be in humans. There have been a couple of cases where they've emerged in domestic pets and some animals of agricultural importance and zoo animals. So I'm going to go ahead and select China and Italy. You can also specify whether you want complete genomes. So that's a nice little feature. And under advanced options, another thing I like to highlight is that you can also remove duplicate genome sequences. So you can remove any sequences that are exact same length and sequence. And I would just like to highlight that our metadata has been standardized. So often when you download metadata from NCBI, you might have, for example, the host with a scientific name, and in another entry, the host might have the common name. So we curate the metadata and we standardize it to make the data sets a lot more searchable and a lot more thorough. So I'm going to go ahead and click on search. and that gives me a nice list of about 220 sequences. And you can sort these by strain name, by the collection date, by the GenBank accession number, by host, and you can go on to either save these to your workbench, you can select them and save your search to workbench for later analysis or add them to a working set. You can also directly run an analysis straight from this page because we like to integrate our bioinformatics tools directly with your search results. So you can perform sequence variation analysis just by selecting these genomes. You can run an alignment on these genomes. And we also have a nice tool which is called the Metadata-Driven Comparative Analysis tool, which I'm going to just highlight briefly. What this tool basically does is performs a statistical analysis based on the metadata that differentiates these groups of sequences. So as I mentioned, I had these two sequences, one from China and one from these two groups of sequences from China and Italy. And you can either separate them manually or you could do auto grouping. And this will kind of group your sequences based on the metadata that's included with them or associated with them. So you can choose either their year of isolation, the virus species, the particular host they were found in. But in this case, I'm going to choose the country. So there are about 220 sequences. You can choose whether you're submitting an aligned sequence or an unaligned FASTA sequences. In this case, I haven't performed an alignment, so I'm just going to keep unaligned FASTA selected and keep my p-value at the default of 0.05 and click continue. So this groups my sequences into 144 from China and 76 from Italy. You can eyeball your sequences just to make sure everything in there is should be in there. If there's anything that you think doesn't belong, you can click on it and remove it, or you can add in case you're missing anything else as well. If everything looks okay, you can just go ahead and click run. And so this can take a few minutes. So what I've done is I've pre-computed this so I can just quickly show you the results of this analysis. So I'm just going to load that from my workbench and scroll down to the results. This shows me a table which includes the position, the chi-square value, the p-value, and degree of freedom. And if you'd like to learn a little bit more about exactly how these are calculated, you can click on the SOP documentation for this tool, which will give you a lot more details. But basically, you can see that there are quite a few differences. So for example, in group 1, which was our group of sequences from China, there's a lot less variation, which makes sense because these were sequences from the beginning of the pandemic. So at this position, 417 in the genome, you can see that most of these positions contain an adenosine, whereas in the sequences from Italy, there is a little bit more variation. So it's a nice way to identify differences in your sequences based on metadata. So moving back to our analysis and visualization tools, we have a couple of other things I'd like to show you. In addition to the typical bioinformatics tools or traditional bioinformatics tools such as BLAST or Primer Design, we have a couple of others, including the Vigor 4 Genome Annotator. And you can use this to either standardize the annotation of your own genomes, or if you have an unannotated genome that you've just sequenced and would like to annotate, you can upload that here, either by clicking on analyze my custom sequences only, or you can just paste in sequences in FASTA formats over here. So this is quite handy because oftentimes in GenBank, you might find one or two sequences that just weren't annotated, or you can find sequences that have been annotated using different software. So you might have the same proteins with different names. So for example, spike can be called surface glycoprotein or spike or surface protein. And it just makes it a little bit more difficult to search when you have to look for three different names. So you can standardize the annotation of your data sets here. And one of the other things that Vigor 4 provides is annotation of the mature peptides. As I mentioned, the ORF1A and ORF1B polyprotein can often just be annotated as ORF1A and ORF1B, whereas the cleaved protein products, the non-structural proteins, are often not annotated. So what Vigor 4 does is adds the annotation for those as well. So just to give you a quick example of how easy this is, you can just copy and paste in any sequence that you want. And I have one as an example over here. And you just go ahead and click run. And again, this can take a minute or two or longer if you have a particularly large data set. So I'm just going to show you the results that I've previously saved in my workbench. And this gives you a table with a few different columns. You can sort it by the start and stop site so that you can see the first open reading frames. And these include the ORF1A and ORF1AB polyproteins. And as I mentioned, we also do annotate the mature peptides, which include the non-structural proteins, as well as the accessory proteins. And something else that we've added, I mentioned that there are a couple of open reading frames in the accessory proteins of SARS-CoV-2 that may or may not be expressed. We've added the annotations for these as well. So for example, ORF3C and ORF3B. ORF3B, there's no evidence yet of its expression, but ORF3C may actually be expressed. And it's not commonly annotated in most of the GenBank records yet. In addition to ORF9B and sometimes ORF9C are also annotated. ORF10 is annotated in most GenBank records, but so far there's been very little evidence of its expression as well. So one final thing I'd like to show you before I turn it over to Richard is our genome assembly and annotation tool. And I've gone into a lot more detail on our Coronavirdae analysis tools in a webinar series that we hosted last autumn, and you can find more detailed and longer videos on our YouTube site. So if you're interested in hearing more about that, please go ahead and look there. So our latest tool for SARS-CoV-2 is the genome assembly and annotation tool, where you can take your raw data from next-gen sequencing reads, or if you like, you can also upload raw reads from the sequence read archive, the SRA database, and use this workflow to assemble complete genomes from raw data, from raw sequencing data. It's hosted on what will eventually be our new site, the Bacterial and Viral Bioinformatics Resource Center, but it's still in beta, but it is hosted here for now. You will need to make an account if you'd like to save your data here, but you can use your ViPR account to log in, and that's what I'm using over here. So your input files, you have a couple of different options of what you can do. This workflow accepts paired end reads. So you can either click on this folder icon, and you can choose to upload your folder. or I have some FASTQ files saved in my SARS-CoV-2 folder. So here's a couple of examples of paired end reads, and I'm going to put the other half here. And then you can click on this arrow to move your, oops, forgot to select the platform. So these are from an Illumina read, but if you're not sure, you can just click on Infer Platform and let the program figure it out for you based on the data. And then you click on the arrow to move the selected libraries, move the reads to the selected libraries for assembly. Moving down to the parameters, if you're not sure which workflow you'd like to choose, you can click on Automatic. If you know you'd like to choose an Illumina or the CDC's Nanopore workflow, all of these, by the way, which are detailed on the CDC's GitHub site, you can choose here. So I'm going to click on CDC Illumina, and I'm going to give my example a label. So test March 16. I have to choose an output folder, and I have a SARS-CoV-2 folder that I've already made for my analyses. And you just go ahead and click on Submit. And if all goes well, you should see this green window come up that says your SARS-CoV-2 genome assembly and annotation job has been queued. If you'd like to monitor what's happening with your genome, you can take a look down here at the jobs, and it'll show you in gray how many jobs you have queued, in yellow how many are running, and in green how many jobs you've completed. And if you'd like to look at an example of one of these types of analyses, you can click on completed job. So I'm going to go ahead and choose this one because often it can take a little time to finish. And this gives you a set of files where you can look at an HTML report that summarizes everything that's come out of this workflow. And it shows you just some of the assembly statistics, what type of library, what workflow you chose for it, how many indeterminate nucleotides were found, and how many blocks of these ends were found in the assembly. This map gives you the coverage depth based on which region of your protein you're looking at. So over here, it looks like the spike area, it's not particularly well covered. And if you scroll down, you can also see a list of the variation data as compared to the original Wuhan 1 reference sequence of SARS-CoV-2. And if you scroll down even further, you'll see that we also provide annotation for your assembled genome based on our Vigor 4 program that I mentioned before. So pretty comprehensive. And if you want to just download the FASTA file for your assembled genome, you can do that by clicking over here and moving to this green column and clicking on download. If you're interested in the annotated genome, you could do the same thing and just click on download. If you'd like to look at these folders called annotation and assembly, it gives you a little bit more information, as well as provides a GenBank file and an EMBL file for submission to NCBI or EMBL databases, as well as a couple of other files you might be interested in, and as well as the complete Vigor 4 output files. So going back to the assembly folder, this contains some more detailed files on the assembly, including the assembly detail, the FASTA files, and the variant calling files as well. Again, I've gone over this in a previous YouTube video, so if in more details, so you are free to reference that, or you can click on our user guides, which gives you a little bit more of an overview and some details on exactly what you should do exactly what software is used for each step, for example, read trimming, read mapping, or variant calling. And I think I'm gonna take a break there and make sure I leave some time for Richard to go over our other sites.
Great, thanks,
Other
Anna.
features.
Yeah, so what we wanted to do is end by showing you a feature that we've implemented over the last month or so, and that is a system to track these emerging variants for SARS-CoV-2. If you go to the ViPR homepage, you'll see at the very top here is a banner that kind of announces this new component, the SARS-CoV-2 variant and lineages of concern. And so if you click on that link, then it takes you to this page that we start out by showing kind of an overview of 5 different lineages that we're currently tracking. The B117 that originally emerged in the UK, the B1351 coming out of South Africa, the P1 lineage coming out of Brazil, this California variant that originated in Southern California, and then B1375, which is another lineage that people are interested in tracking. And so, on this overview page, we have some just basic information about these lineages. They have different lineage annotations coming from either Pango or NextStrain. They have some other synonyms that are sometimes used to refer to these lineages. Information about the emergent date and location, and the key amino acid substitutions in the spike protein that kind of characterize these different lineages. And then finally, the reason that we're interested in tracking these is because there's experimental and/or epidemiological evidence to suggest that these lineages have important characteristics related to increased transmissibility or the potential impact that they would have on some of our diagnostic tests or some of the therapeutic antibodies that are being used to treat patients with COVID. And then on this next tab, we have each of the individual lineages. We have a lot more detailed information on this tab. So for example, in addition to the amino acid substitutions in the spike, many of them also carry amino acid substitutions in non-structural proteins or other structural proteins. So we have listed here all of the amino acid substitutions, as well as all the nucleotide substitutions in comparison with the Wuhan HU1 reference strain. Down here at the bottom, we have links to representative strains for each of the lineages. So if you click on this link, this takes you to the ViPR page with detailed information about this representative isolate for, in this case, the B117 lineage, including all the annotations coming from Vigor that Anna just described. And then we also have computed consensus sequences for the lineage that are also available here. And if you're interested in how all this information was determined, there is a detailed document, an SOP document that describes exactly how the sequence variations and consensus sequences were determined, and that's all described here. So this is kind of information about lineages that have already been identified that we want to keep track of, but we also are trying to monitor the emergence of new lineages as they're emerging in real time. And so in this covariant tab here, what we're doing is we're basically looking at the sequence records and quantifying the frequency of particular amino acid substitutions in the spike protein and trying to monitor how the frequency of those sequences might be changing over time. And so this is an example of really the kind of the top 10 lineages that have been observed worldwide. But you can filter this list using different criteria. So for example, if I'm only interested in, so this is looking at all regions over all months. But if I'm interested in maybe just looking at what's going on in the UK or the US, I can kind of change the filter. So if you just notice here, worldwide, the most frequent lineages are the B117 lineage that kind of is emerging out of the UK. But if I change this to look at just the United States, you'll see that there's a different picture emerging, where here we see that although B117 is in the top list of variants that we see in the US, this California variant is actually the one that's emerging as the most common variant that we're seeing in the United States. Over here, we're actually monitoring the growth rate of these different lineages. And in the most recent month, this is just kind of tracking what has happened in the last month. So there's really been no increase. But what you can do is you can change the filter for a month. And if we go back to, say, November of last year, and we change the filter, now what you can see over here is that there are some lineages that have been increasing pretty dramatically from month to month. And in fact, this was around the time when this California lineage was emerging. And going from October to November, the increased frequency of sequences that carry these substitutions increased by 25-fold. And so this is kind of an indication of a new emerging variant that seems to have increased fitness, and its frequency is increasing dramatically during that time period. Now, if we were to go back to and look at what was going on in the UK in November, you'll see that there was a different scenario. So in the UK, we see that there was a really high increase in the growth rate of this lineage, and this was the B117 lineage that emerged sometime in the autumn in the UK. And so this is evidence to support the idea that it's really increasing in prevalence in that region. So anyway, there's a lot of information here that you can explore in terms of what's going on in different regions of the world at different times. We also, in addition to this table, we have some precompiled charts about the emergence of the different lineages. And so in this case, this is in the United States, lineages that are kind of co-circulating right now. And in gray here, you see this is this California 20 lineage that came out of Southern California. So this is the pattern of what we're seeing circulating in the US right now. But if you are interested in what's going on in UK, in contrast, you can just change the country, and you see that in the UK, there's a very different picture. And this green lineage here that is really taking over in the UK is the B117 lineage. So this is like tracking the covariates, or the lineages that have certain constellations of substitutions. But you can also look at individual substitutions separately. And so that's in the variant tab. And over here, you can see these are kind of the top variants that are present in the coronavirus genome sequences. And the most common, most frequent single substitution is the D614G substitution that emerged pretty early on in the evolution of SARS-CoV-2 and really has become predominant. So virtually all of the lineages carry this substitution in addition to other substitutions. One of the nice features that you can do here is if you're interested in a particular variant, you can use the keyword search. And so one of the variants that people are starting to track is substitutions at position 677 in the spike protein. And so I can kind of change this filter here. And so what you can see here is that there are actually 3 different substitutions at this position that have been emerging and increasing in frequency. Right now, this is for the most recent month. But if we go back to, again, if we go back to, say, last month, change the filter, or the month before, Now you can see that indeed all three of these lineages were increasing in growth rate in January. And this is particularly interesting because this is adjacent to the furin cleavage site. And we think that at least some of these substitutions may be increasing the efficiency of furin cleavage of the spike protein, which could then impact, infection efficiency. The other thing that we've been doing is we've been trying to compile a list of interesting sequence features associated with the coronavirus. And we get this information from a variety of different sources, but here you can see at the top the different open reading frames and mature peptides. And then over here we have information about enzyme active sites, other regions of interest like the ACE2 binding region for the spike glycoprotein, the different domains that have been characterized in the different open reading frames, information about experiments in which a mutation affects the property of the virus or the proteins in those regions. And then down here are the different substitutions for the different lineages of concern. We also have information about the location of antibody epitopes. So the goal here is to try to allow people to look for the the overlap between mutations that are occurring in different lineages and interesting information about what's known about functional regions or antibody epitopes that might be also found in those same regions. And so with this genome browser, you can kind of zoom in. And so I'm going to zoom in here on the receptor binding domain of the spike protein. And now you can see a little bit more detail about what's going on here. One of the most recent things that we added to this genome browser is Jesse Bloom's group from the Fred Hutchinson Cancer Research Center recently reported a series of experiments where they looked at the impact of mutations within the receptor binding domain on the ability of a variety of different antibody preparations to bind to the spike protein. In this case, and so you can choose to, add new tracks to this genome browser. And in this case, this is information about the impact of mutations on polyclonal plasma serum or plasma antibodies from infected patients to be able to still recognize the spike protein if there's a mutation at this site. And so this information we're then using to track variants that might actually impact the effectiveness of vaccines that are currently being administered to people or natural immunity that's developed in infected people. So if we start to see substitutions at this position, for example, here, it might raise some concerns that we're detecting antibody escape variants emerging that could impact vaccine efficacy. So anyway, there's a lot of different information that we're bringing in to allow for kind of looking for overlap between mutations and important properties of the virus that have come from a variety of different sources. This resource is continuing to grow. We're going to be adding a 3D protein structure viewer in the near future, a phylogenetic tool, et cetera. This is going to be, I think, a really useful resource for tracking the impact that emerging variants are having on the properties of the virus. So I'm going to stop there and maybe take a few questions if there are any before we introduce our guest lecturer.
So Richard, there was a question about how the lineages are named and is there any meaning attached?
Mm-hmm. So there are two main resources that are naming these lineages. One is called Pango, that's coming out of the UK, and then the next strain lineage is coming out of the United States. With the Pango lineages, there is some information about kind of the how these lineages are emerging, like what the parent strain was that the lineage emerged from based on this nomenclature. But one has to really take that with a grain of salt because it's not really a strict hierarchy. There are definitely exceptions to the rule, but they are trying to capture some information about kind of ancestral lineages from which these new variants are emerging.
Okay, so I don't see any more questions. So if you'd like to go ahead and introduce Duncan, I can make him a presenter in the meantime.
Okay, yeah. I see that Duncan is on the call, so it's a real pleasure for me to introduce Duncan McCannell, who is at the CDC in the Division of Advanced Molecular Detection in the United States. Um, Duncan, if you don't know Duncan, Duncan is playing a really important role within the public health community right now because he's involved in coordinating all of the sequencing efforts that are being done by the public health community, both in the state and local public health laboratories, so that we really understand how these variants are emerging and how their prevalence might be changing over time. So he assembled what's called the SARS Sequencing for Public Health Emergency Response, Epidemiology, and Surveillance Consortium. And Duncan, correct me if I'm wrong, but I think there's about 600 people who are involved in the consortium now, maybe more. But it's
We
an
topped 900 this week.
oh wow, that's
It's gotten
crazy.
big.
Yeah. So really, this is the group that is at the front lines of determining the genome sequences for coronavirus that's circulating in the United States. And really, the idea is that we want to monitor the important changes that are occurring in the genome sequence and trying to predict what impact they might have on things like transmissibility, the effectiveness of diagnostic tests, the effectiveness of therapeutics and vaccines, and also to help support contact tracing and other kinds of epidemiological analysis and interventions that we might want to use in order to kind of curtail the spread of some of these emerging variants. So Duncan is really single-handedly kind of herding the cats, as we would say, in keeping these sequencing efforts kind of on track and providing the kind of information we need to monitor how the virus is evolving. So Duncan, we're really pleased to have you join us and looking forward to your seminar.
Thanks, Richard. And I think I can probably take credit for herding some of the cats, but it really is a multi-pronged effort. There's many people across CDC, there's many people across state and local public health departments that are working in genomic surveillance right now. And one of the real fundamental changes, one of the fundamental differences from a lot of our conventional surveillance and outbreak response has been just the sheer involvement, sheer scope of the involvement of academia and the private sector in this effort. It really is unprecedented, and the volume of data that's being generated, the types of data that are being generated is new and exciting, and potentially presents some exciting opportunities for not only this pandemic but future infectious disease challenges. Today, I wanted to go into a little bit of a history of genomic surveillance in the U.S. I've warts and all. I will try and be, I think it's good as we sort of approach the 1 year mark of genomic surveillance efforts within the country to stop and take stock, to look at where we are and where things are currently going, and to give you a sense of what we're currently starting to see from the various activities that are spinning up. So a good place to start is with my program for people that aren't familiar. I work in an office at CDC that traditionally has been fairly small. We are the Office of Advanced Molecular Detection, and our mandate, we came into being about 7 years ago when the agency realized that technologies like sequencing were suddenly becoming feasible to do at the kinds of scales, the types of applications that we needed for public health. But at the same time, we didn't necessarily have the necessary laboratory and bioinformatic infrastructure within public health. We didn't have the workforce components that we needed, and we needed to start addressing these if we were going to effectively bring these to the forefront of public health application. Public health isn't always known as a center of innovation. We usually are not the absolute bleeding edge, and much of that is by design. A lot of the assays that we're trying to run, we're running at very large scales, and we need them to run reliably and reproducibly and across a wide number of labs. And so, I think our office has tried to push an innovation program as much as we can, and we will continue to do that as we go forward. A lot of our focus has been on how to operationalize these technologies, how to standardize them, how to roll them out in consistent ways. There we go. So just to give you a sense, if we go back to 2013, my slides are reminded they're on this morning. Really only a handful of state public health labs had sequencing capacity, about 5 or 6. A lot of that was driven by FDA's genome tracker and foodborne disease detection. And in many of these labs, the sequencing instruments themselves weren't actually located at the public health labs themselves. They were at a local institution or academic partner that could actually house and support these kinds of instruments. Fast forward just 5 years, it took a little bit of time to eventually roll that out, but as of 2018, every state public health lab in the country now has sequencing capacity. A lot of that is still focused around some specific applications like foodborne disease detection and flu are 2 big ones. But increasingly, public health labs have been using that kind of capability, that kind of capacity to drive additional applications. Healthcare-associated infections and investigations of viral hepatitis are frequently used. Legionella, tuberculosis, there's quite a range of different applications. So really not surprisingly, we sort of expected that that many public health labs, when the pandemic hit, would there would be an initial response period during which they'd adjust to the new workload and the demands of the pandemic. But many were in a position where they could apply genomics as part of their response strategy, as part of their investigation strategy. And I think you'll see we've had to make some adjustments as we've gone along. Sorry for my dog. One of the other challenges is workforce development. In many of these public health departments, there isn't necessarily a deep bench when it comes to genomic expertise or bioinformatic expertise, and this can present some universal challenges. It's something that we see at the federal level. It's something that is difficult to replicate and extend at the state and local level. So shifting gears, so why would you want to sequence SARS-CoV-2 at scale? I think going over some of the demonstrations that Richard just went through, and Anna's presentation earlier, I think a lot of these become self-evident. First of all, you want to be able to monitor viral diversity over time. You want to understand, gosh, I keep on hitting buttons today. Understand changes in transmission dynamics. Those are both local as well as regional and national. But ideally, you want to be able to translate this into effective public health responses. So can you use it effectively to help drive contact tracing and your overall containment or mitigation strategy? To do that requires a lot of moving parts to work together very fluidly. You need to have, and I'll go over this in a minute, but you need to have data generation, you need to have testing all sort of fit together nicely. You have to have sequencing in a timely fashion, bioinformatics to actually analyze and interpret the data, and more importantly, that has to be integrated back into the public health information flow and acted upon. Those are all challenging pieces to put together at almost any level of public health, and certainly have been a challenge in a lot of jurisdictions throughout the world as they try to grapple with this. There's also, obviously, as anyone who has read the news lately, a lot of concern over the emergence of clinically important variants. So these could be things with unusual virulence or phenotypes. These could be variants that potentially disrupt a diagnostic or therapeutic agent or have disruption to their antigenic targets. These are all things that we're closely monitoring, and with the emergence of variants has become definitely part of the forefront. And as we roll out vaccination campaigns, I think it's critically important for us to be able to monitor these population level changes in phylogenetic as we apply vaccine pressure and monitor for vaccine escape and assess those variants as they come up. One of the things that has been missing historically across the US has really been sustainable and longitudinal data collection. There's been many parts of the country where we just have not had consistent sequencing for a variety of reasons. On other parts of the country, we've had very deep sequencing or focused sequencing on outbreaks or other things. So, sometimes looking across this and trying to really get a national picture has been a challenge. But throughout all of this, again, I think one of the opportunities that shines through is the opportunity to use these events as an opportunity to build new collaborations and to help drive innovation around public health. And I think that's something that we're trying to do in various regards. So let's look at sequence generation so far. This is as of last night. But one thing that we have been pushing from the CDC perspective has been the submission of sequence data, not only to GISAID, the consensus sequence to GISAID, but we think it's also really important to submit the raw sequence data to SRA and sequences to GenBank. Part of the reason for that is to enable platforms like ViPR that aren't necessarily able to operate within the confines of the existing GISAID data use agreements. It gives a lot more downstream flexibility in terms of how these data are used. And from our perspective, if data are being generated as part of federal or state public health response, those data are generated with tax dollars. They should be available as broadly as possible and as to the maximum utility of the American public and the global public. As of yesterday, we have about 84,000 sequences in GISAID, about 81,000 sequences in GenBank at this point. I don't have SRA statistics, although NCBI has been promising me an easy way to survey that for a while, so hopefully I will get that soon. But you can see that there's been significant acceleration. It took almost a year for us to hit 60,000 sequences. We've been doing about 60,000 sequences a month, or for the past several months. So it's accelerated extremely quickly over the past little bit, and the detection of variants, the acceleration of the national surveillance program has really been a part of that. You can see some of that in the total proportion of cases. I mean, as of now, if you look at raw numbers, we're at 0.62% of total U.S. cases sequenced. A lot of that is a denominator issue. We've had a lot of historical cases in the country. If you take a rolling average, though, of the past couple of weeks, we're at 500,000, 400,000 cases a week. So we're actually hitting fairly decent proportions of the total number of national cases and starting to get really good data visibility. So let's talk a little bit about the early days, taking back to February or March of 2020. As I said, I think one of our expectations was that state public health labs, local public health labs would have the initial shock of the wave of cases that would hit, but that many had sequencing capability in-house, and many would start to sequence as part of their response activities. If you look at submissions to GISAID, however, it tells a little bit of a different story. In the first couple months of the first half of last year, the majority of sequences were not being generated by public health labs. They were being generated by academic labs, by clinical labs, and a variety of other sources. The number of sequences from public health labs started to pick up in the second half of the year, but really it was pretty uneven in terms of those submissions. You had a few public health labs that were submitting full tilt. You had many others where just did not have the capability to get up and running in any sort of meaningful way. So at the end of April, actually at the end of March is when we soft launched it. But at the end of April, we announced what became the SPHERES Consortium, or Sequencing for Public Health Emergency Response, Epidemiology, and Surveillance. Our goal here really was pretty simple. We didn't necessarily have, we anticipated that we would have a national genomic surveillance strategy down the road a few months. But in the meantime, it made a lot of sense to try and gather all of these different sequencing efforts, independent sequencing efforts, to align them as much as possible with federal, state, and local public health, to ensure that the data was being submitted to public repositories as quickly as possible, and to start talking about standardized ways of doing that. We didn't want to necessarily micromanage the sequencing process, but we did want to see if we could improve the quality of the output of all these different sequencing labs and create really a community of expertise around this. And so with that, Spheres was born. We started fairly modestly, as we said at the top, though. Right now, we're at over 900 scientists from over 200 organizations. This really is one of the most ambitious mixes of public and private sector that I think CDC has attempted. And it brings together a lot of the state and local public health departments, a lot of county and local public health departments, I said local twice, and a lot of federal agencies that are involved in these activities. But more importantly, a lot of the private sector, a lot of academic partners, all essentially focused on fairly common goals. There are various tribes that have started to emerge within Spheres. We have ones focused on environmental and wastewater surveillance, others that are focused more on clinical testing, just to give two examples. But the through line from that is really a commitment to large-scale sequencing, to applying sequencing as part of a strategy for response and outbreak detection. Let's talk a little bit about why sequencing is hard and sequencing surveillance is hard. One of the challenges that we have in the United States is that our testing methods are really quite distributed. If you think about any municipal area in the country, there's multiple private labs that conduct the majority of testing. And none of them are necessarily incentivized to hang on to those samples or to transmit those samples or data onto public health or a lab that can sequence. So in most cases, in virtually all cases, sequencing capacity is really not the rate limiting step. It's access to those samples and ensuring that you can do that in a timely fashion. This piece here in the middle here is really the core of the AMD program itself, and something that we've been working on for a long time. How do you effectively integrate sequencing activities, epidemiology to interpret it, and the bioinformatic expertise and resources needed to actually turn these data into actionable intelligence? That's challenging to do at the federal level. It's definitely challenging to do it at at state and local level, especially if you're trying to do it quickly and to try and turn those information into action. In the case of SARS-CoV-2, if your turnaround time is 2 weeks or 3 weeks, you're looking at an outbreak investigation very far in the rearview mirror. You really need to have really rapid data turnaround, and sometimes having all those necessary handoffs, even if you have the partnerships in place, is extremely challenging to get that in a timely fashion. So as you think about this, though, there's a lot of factors that come into it. Turnaround time, as I said, is a big key piece of it. The cost and sustainability of this kind of effort is also key, and that will inform everything from your sampling strategy to where you source your samples from to how you actually approach sequencing overall. Through the whole thing, you need to think about the actionability of the data. It's very easy to go out and sequence hundreds or even thousands of samples, but not necessarily get any meaningful insight from that. So you have to really ensure that you have a good system for prioritization and that you are focused on what the public health outcomes of those data should be and the insights that you're trying to derive. That's going to inform things like how you scale and how you select those objectives. But one of the big challenges that underpins all of this really comes down to logistics and data sharing. It's how do you actually get the swabs from the patients, get them tested, get them routed to sequencing, and get both the sequence sample and the data connected in a timely fashion and in ways that can actually be useful. There's a lot of challenges, as I said. Again, diagnostic testing is conducted by various private sector labs, which present some challenges when you're trying to connect those with samples. Public health labs right now are stretched extremely thin, so the ability for them to bring a deep bench of epidemiologists and laboratorians to these kinds of challenges is often very limited. Epidemiologists, one of the challenges we have across the U.S. public health system, across the global public health system, is that genomic epidemiology, molecular epidemiology in general, is still a fairly limited set of expertise in many public health departments. So, that's an area where we have to make some focused workforce development, because it's one thing to generate these types of data to make them available. If you can't actually interpret them and put them into action and actually apply them to public health response and policy, it can be a very expensive and fruitless exercise. The other thing is, the sequencing data is not always as informative as it could be, and you have to kind of know selectively where you can apply it. In the case of SARS-CoV-2, for example, it's a virus that doesn't have, since it does have proofreading capability, it doesn't have an extremely fast molecular clock. And so if you're trying to understand chains of transmission within a very fast-moving outbreak or within a congregate setting, it may not give you additional insight beyond what perhaps even PCR testing could give you. It's important to understand the limitations of that and to communicate that effectively, and to know when you're going to likely get the most bang for your buck. So our strategy from CDC has really been to focus on 3 different levels. Initially and ongoing, we've been focusing on the Spheres network. I think our goal there is to really establish this as not only a community of practice, but transition this into a broader centers of excellence academic model over time. We focused on 2 different levels, though. One on the national level, which is what I'm going to talk about mostly today, but probably some of the more exciting impact is really at the state and local level. We've got a new team that we've stood up that is really designed to help facilitate this in state and local public health departments to build on some of these academic partnerships and to help bring standardized analysis and reporting into the hands of more public health authorities across the country. So I'm going to talk about 2 different programs at the national level today. One is NS3, or our National SARS-CoV-2 Strain Surveillance Program, which started off in November of 2020. It's a very traditional public health surveillance model. So initially we were looking at over 20 random positive samples per month per jurisdiction. This is essentially how we surveil for seasonal flu right now using conventional models. The advantages of this is, we started out at this level. We've since scaled it up to about 3,000 sequences or samples a month, or specimens a month. The advantages of this, though, are that not only that we're sequencing these centrally, so all these specimens from 64 public health jurisdictions, so all 50 states plus a lot of territories and localities, are coming into CDC for standardized sequencing and analysis. But more importantly, those viruses are available for characterization and for banking and for additional workup. So that's a critical distinction of that. The other piece has been just trying to scale things up with sequencing generally. A lot of the country is difficult to surveil. It's difficult to surveil from within the public health lab system. Really we've had to get a little creative with how to bring the sequencing really to the samples. What we've done here is actually started a series of contracts that we call baseline contracts, working with some of the large commercial diagnostic labs, since they do have access to essentially real-time samples from a really broad geography. And what we essentially did is just looked at diagnostic test volume nationally, looked at the geographic coverage of these different laboratory networks, and then evaluated which ones could incorporate sequencing as part of their workflow. This has allowed us to really scale up sequencing in large parts of the country where we didn't have good data. It's allowed us to, in the very early days of variant detection, it's allowed us to apply a national filter to do S-gene target failure surveys to look for B117 in particular. But now these contracts all essentially do random surveillance for the entire country and are pulling in thousands of sequences a week. All 3 of these, though, do work together. So we've got Spheres, which has participants from across the country, institutions from across the country that are working on a variety of different applied applications and surveillance and outbreak response. We've got NS3 and Baseline, which are really designed to paint broad stroke genomic epidemiology across the country and give a sense of what's circulating and where. And then we have this focus on building out state and local public health response and their ability to actually apply these much closer to the ground. And these are connected by ensuring that we have consistent data, consistent metadata flowing into multiple public repositories as quickly as possible. If we look at the data, so this is out of GISAID. It gives you a sense of just what is currently being deposited across the US. These data are a little bit old, but I think they're from March 12th is when I took this screenshot from one of our dashboards. But it gives you a sense of overall submissions. And you can definitely see the down here in the bottom right, when national surveillance really started to kick off, a lot of the data started to come in very, very quickly. Sorry, just 1 second. One of the other things that we've made available has been a series of next strain builds. So if you, oops, screen. If you go to nextstrain.org/group/spheres, this is data from our NS3 surveillance, and it essentially is focused subsampling. It gives you a sense of those samples that have come in so far. We've got almost 1,500 sequences in there, and it essentially gives you a running tally of samples that have come in through the door that way. One of the other things to add is that we actually have state-focused builds for each one of the states as a starting point for molecular epidemiology. If you go to nextstrain.org/group/spheres, each one of these are built essentially every week on Tuesday nights usually, based on all the sequences that are currently in GISAID. For each state, so this is California here, you can see that some states have been sequencing very regularly throughout the pandemic. So the subject state or query state is always in red. Other states are usually in different colors, and global context is usually in gray. So it gives you a very rapid way of comparing not only what is circulating in the state, what is coming up during in essentially the GISAID collection, but also what is some national and global comparators to it. You can also see the effects of the surge. Here's an example of Missouri, which didn't necessarily have the same sort of robust genomic surveillance throughout much of last year that California did. But you can see with the ramp up of some of these national programs and the increased sequencing output of the Missouri lab, suddenly a pulse of sequence data that will hopefully continue on into the future. So one thing that often comes up is, how do you actually classify variants? And this actually is brand new as of this morning. We just updated the variants page. We've classified and provided criteria for what we could deem variants of interest, variants of concern, and variants of high consequence. So variant of interest is essentially based on genotypic features. So specific genetic markers that have been associated with changes in receptor binding or reduced neutralization. These are mostly based on feature analysis of the sequences themselves. The other two, so variant of concern and variant of consequence, really are based on the functional consequences of those mutations. So where there's actual evidence for increased transmissibility or more severe disease, where there's a significant reduction in neutralization antibodies or in frontline therapeutic agents. And some of the examples of these, are the Pango lineages that Richard just covered. So B117, P1, and B1351 are the three recognized currently. We are in the process of elevating B1427 and B1429, which is the L452R variant first identified in Southern California, as a potential variant of concern, mostly because it does show evidence of increased transmissibility, but it also has a fairly significant impact. The L452R mutation has very significant impact on one of the monoclonal antibody frontline monoclonals. Variant of high consequence is essentially the highest tier that we have currently. We felt that it was important to have something above and beyond concerning to really indicate that those variants that had very clear evidence that existing prevention measures or medical countermeasures were failing routinely. So there are no currently identified in this way. This essentially adds another tier on top of the current variant of concern, but we felt that it was important to have some escalation beyond what we currently had. So this is from the Anderson Group's excellent outbreak info resource at Scripps. It gives a good overview of some of the variants of concern. Actually, I guess 5 of these would now be variants of concern, and B1526, which was recently identified in New York. One thing I do want to highlight for your attention, though, is that a lot of these variants of concern are long branch mutations. So they emerge. They have a lot of compound mutations and covariation that we see especially in and around the spike gene. And these are fairly characteristic. They allow us to identify these fairly routinely, and that is a useful feature. If I go to the sequence data from our commercial contract, so this is from actually this week. This brings in data from 3 commercial contracts. So gosh, sorry, I'm trying to bring up the dashboard at the same time. If I can do that. Work? Yes. All right, live view. So this is actually data from LabCorp, Quest, and Helix and Illumina. So LabCorp provides national testing. LabCorp and Quest both provide national testing for a variety of different labs. Helix provides residential or municipal testing within the San Diego County area, but also provides back-end testing for most CVS pharmacies across the country. This gives you a sense of the overall breakdown over time. We've had just over 30,000 sequences that have come in through these contracts as of last week, and that's since the mid-December period. So it gives you a sense of the types of volumes that we're seeing. This is a little bit lagged. In terms of breakdown, so this gives you a sense of overall proportion. In pink here is the B1427, B1429. I would, these are raw proportions and aren't necessarily, I would not take them as prevalence estimates just because there is some weighting that needs to occur based on test volume and such. But it gives you a sense of where we're seeing the distribution of some of these variants across the country. So B1427, B1429, that's the so-called California variant. We are seeing it in virtually every state at this point, so it is fairly broadly disseminated, with a center of gravity out in the western states. If we look at the 3 other variants of concern, so B117, again, you see fairly distributed cases. If you look at the CDC data tracker, which I'll show in a second, we have reports from the majority of cases at states at this point, with probably the majority of cases up here in the Northeast, in Florida and California, just in terms of raw proportions right now. B1351, out of the contract sequence data, so far we've seen 64 sequences. That is all pretty heavily weighted towards the East Coast, but I would be surprised if we didn't see that more broadly. And I'll show you B1526, which is the variant that was first identified in New York. And you can see that there is, so this is independent of data that was initially reported in GISAID and some of that initial reporting that came out a couple of weeks ago. But you can see that there is some weighting around the upper Northeast, but again, sort of that distribution of cases across the U.S. So again, I think one of the more exciting pieces of this, though, is the utility of the sequence data at the local level. And so we have a technical outreach team called TOAST that is really designed to enable these kinds of outbreak investigations and to put tools into the hands of more state and local public health departments to actually do these kinds of investigations. This is a long-term healthcare facility outbreak from Minnesota. It gives you a sense of, again, many of these viruses end up being fairly indistinguishable just because of the time frame and the nature of these outbreaks. But it does give you some ability to use this as an investigation tool and understand introductions into a facility, or when you are seeing breakdowns in infection control or other potential public health or clinical interventions. Concomitant to this, we've recently put out a genomic epidemiology toolkit. This is a modular example or case study driven toolkit that has both case studies as well as examples of how you can implement. So it's a walkthrough of how to use tools like NextStrain and MicrobeTrace. It provides some worked examples of different outbreak investigations and what this has been useful, and also gives a great overview of things like variants and how those are interpreted. Right now, it's essentially short, bite-sized modules. We've tried to make it as accessible as possible, and we've tried to make it as flexible as possible. We're adding content as we identify areas where it makes sense to do so. We're trying to make sure that we're being as responsive to the broader community as possible, but this is a new resource that we're pretty excited about. I'd also like to draw your attention to the National Genomic Surveillance Dashboard. I'm not going to show it today, but we're updating that several times a week now with numbers, with maps of what we're seeing and where. It's a lot of that is going to transition into the COVID data tracker and some of the other tools that we have for data availability. So it should be much more scrapable in the near future. I talked about this earlier, but the SARS-CoV-2 variants page was just updated. It's got some great examples, some great explainers on how variant of interest, variant of concern, and variant of high consequence are delineated, some examples of each and key mutations. It also has links to reference material. So the BEI references are all linked now to each one of at least the 3 initial variants of concern. And we'll continue to update this and expand it as time goes on. And lastly, this is the dashboard that I talked about. This gives you a sense of how many jurisdictions are currently reporting various variants. You can see for B117, it's virtually all states and localities. So that's probably the best definitive resource. Those include not only sequences that are in some of the public repositories, which again can be subject to bias, but these are usually, everything that's on here has been EPI confirmed by the state and local public health department. So it probably is the most definitive source of information. And with that, I'll open it up to questions. I realize I'm a little over.
Great. Thanks so much, Duncan. That was terrific. Well, maybe I'll start off with the questions. So, toward the beginning of your talk, you emphasized the importance of actionability on the genomic sequence data and the other information that's being kind of pulled together. And I'm just kind of curious, like, where do we stand in the United States in terms of using information about emerging variants to do contact tracing and quarantining and things like that? I mean, I've seen some presentations about, what happens in China and Hong Kong. They have a pretty serious contact tracing quarantining system, and it seems to be quite effective in limiting the transmission of outbreak strains. Where do we stand in the United States with that kind of work?
That's a good question. And I think, the short answer is it varies by jurisdiction. Some jurisdictions are very well positioned to be able to work on these kinds of data. Others are not necessarily in a position to do so. I think even in jurisdictions where the capability exists, case rates have just been so high until fairly recently that it hasn't necessarily been a primary consideration. It's been key, but general guidance around a lot of these variants is that the effective non-therapeutic interventions don't change. It's still masking, it's still maintaining social distancing. It's a lot of those fundamental pieces still apply. I think as case rates start to come down and we're out of just sheer firefighting mode into more, following flare-ups and outbreaks, I think it will become a much more important tool in many places. But I think a lot of that is really going to be driven on technical capability. The ability to actually do this fast enough to be in a meaningful time frame. If you're generating data a month out, it's useful academically. It can perhaps give you some policy guidance that can be applied to future outbreaks or prevention, but it's not necessarily going to help you in the moment. And to actually turn data around fast enough to help guide contact tracing is extremely challenging.
Yeah,
A lot
it's
of
a
things have to
challenge.
go absolutely completely right.
Yeah, yeah.
So I'd just like
Looks
to take a moment to invite people to, you are able to unmute yourself and ask your question in person. If not, feel free to use the chat to ask your questions. There is another question that says, do you see this consortium being used for other diseases in the future?
I think so. So one of the maybe not this consortium per se, but I think it is a model for us that has been extremely successful. What we are looking at doing is expanding essentially genomic surveillance writ large. There's a lot of commonalities between different infectious disease surveillance tools. There's some of the expertise can work across multiple different pathogens. A lot of the critical platform needs are the same. Whether you're talking about a foodborne bacteria or whether you're talking about vector-borne disease, you still need the basic infrastructure to do those types of analyses, to share data, to integrate metadata, and to handle the sequence data writ large. Making sure that we have flexible tools is an exciting thing. In terms of the consortium, one of the most exciting things is just the number of new partners that it has brought to the table. Now, granted, these are extenuating circumstances. It's one advantage of a pandemic, if there is an advantage to a pandemic, is that it focuses the broader research community on a specific pathogen, with laser-like precision. We're hopeful, though, that a lot of this can be applied to different other pathogens, that we can use a lot of these new ideas, these fresh perspectives, in ways to really reinvigorate public health and to not create these one-off siloed solutions that often permeate the public health space.
Right. Okay, so there's another question on, are there any variants of concern also for sustainably crossing species barrier?
I think it's certainly been reported. I mean, if you look at some of the mink-associated outbreaks that have been reported in Denmark and elsewhere, a few of the key mutations, like the N501Y, N501T, have been associated with adaptation to other host species like rodents and mink and others. How much of a concern that's going to be, I don't really know. One of the early speculations was, whether or not this would lead to new potential vectors or new potential reservoirs. I think that remains to be seen. But one thing that is very nice is that this, as part of our U.S. government risk assessment, we do have a fairly large One Health group that is taking a very holistic view of SARS-CoV-2, taking a look at a lot of the animal health considerations, and trying to really bring a lot of that data in parallel with human clinical disease. So I think it's something that we're definitely considering closely.
And so there's a related question on whether there are any efforts to consolidate SARS-CoV-2 sequence data from non-human animals. And I do believe the USDA is doing that, so I'm going to post the link in the chat. But feel free to answer that as well.
No, I think that was where I would have pointed them.
Okay.
You can search for that in some of the repositories. Like I say, it does have host species, GenBank, and NCBI certainly do.
Mm-hmm.
I think most of them are fairly representative there, but.
Okay, and so another question is, do you see any changes in variant distribution since the vaccine is rolling out?
That is an excellent question, and that is something that we are closely monitoring. I think one of the challenges has just been getting large enough data to actually be able to monitor some of these trends. I think we're continuing to ramp up. Again, we started from a little bit of a cold start. We got a late start to the game. But I think one of the nice things is we do have a few months of data in advance of any vaccines rolling out in any meaningful way in most jurisdictions. So we'll be able to monitor for some of these escapes. I think we're already seeing, definitely convergence of some sites that are obviously under heavy immune selection. I mean, E484K is a good example of that pops up fairly frequently. I think looking at those as vaccines continue to roll out, though, is going to be critically important. But part of that is going to be getting sufficient numbers to actually be able to do proportion analysis and trends and look at vaccine failures and have enough data to actually make meaningful comparisons.
Okay.
And Duncan, I had another question to follow up on one of the questions about pivoting the Spheres Consortium to new infectious diseases. One of the viruses that we've worked on in my lab is the virus D68, and I don't know how familiar you are with that, but in some cases it causes acute flaccid myelitis in infected kids. And in the past, it's been on a biennial cycle, and 2020 was supposed to be, an outbreak year, and we didn't see that probably because of all the, social distancing and all the efforts to mitigate, interactions because of coronavirus. And so we're actually speculating that 2021 may actually be a pretty big year for D68 because now we have a larger susceptible population that wasn't infected in 2020. It'd be really great if, typically those outbreaks start in August. And so it would be really great if we start to see cases emerging in August to be able to utilize, this infrastructure that's been developed in the public health community to be able to pivot to, getting good sequence data on D68 if it does emerge, because that's something that was kind of lacking in the past. So, it's something worth thinking about and maybe planning for, being able to, incorporate some of those samples into the whole program.
Yeah, I think our goal is to not only help motivate the community, the broader community, and bring in more academic participation in the public health process, but again, to build some of these platforms that will enable rapid data sharing, rapid analysis, and comparison. In thinking about that, we are very much taking an eye towards how to ensure that they're maximally interoperable and maximally open so that they can take advantage of existing platforms and capabilities like ViPR and plug essentially everything that we're doing in public health surveillance into those downstream tools. I think it's for spheres, I don't think we're necessarily looking to pivot the entire community over to every single infectious disease. We realize that some, it's an interesting group because some are have tons of experience and roadmap in the virological space. Some are very newcomers that have just had sequencing capability that have some ability to actually do that and have made important contributions to the overall effort. And so I think we're trying to figure out how to manage that, how to shift that. Realistically, globally, we're going to be monitoring, understanding the impact of coronaviruses for years to come. Using this as an applied research community that can help focus on some of these areas, but perhaps broader virological impacts is good. But I think it also gives us a model of these kinds of collaborations and how we can potentially steer them around other broad areas of infectious concern.
Yeah. And I think you emphasized the training aspects of the Spheres Consortium, and I think that's going to have a really long-term impact on the local public health laboratories, being able to respond to local outbreaks. I mean, I think it's really going to have an important impact down the road.
One of the most impactful things that I think the AMD program has had so far, or launched so far, has been our AMD Fellowship Program. So we have about 10 fellows per year. They're masters or postdoctoral level. We place them at either state or federal public health labs. So they're usually at a CDC program or in a state lab. We pay a lot of very close attention to mentorship opportunities and resources and trying to make sure that they're successful. But many of these are people with bioinformatics expertise that have a lot of these new skills that are in demand. Because the reality is, we can't compete with the private sector for a lot of these skill sets. Bioinformaticians are still, they can go to industry and probably make significantly more. What we can do though is create these fellowship opportunities, and what we found is that a lot of our fellows stay around and have actually built the foundation for a lot of our public health bioinformatics community. So we now have a fairly thriving community of practice. We call it Staff B, or the State Public Health Bioinformatics Group. And it's surprising how many of those are either former fellows or former mentors of fellows, or it really has become sort of a self-perpetuating thing that really has started to build out the type of workforce that we need. But it's been a phenomenal change over the course of 5 years. And I think last time I looked at retention, I think we have 70% of people that come through the fellowship program stay on in either federal or state public health. So it's been a fantastic way of doing that.
Yeah, it's great.
If I could ask just one more question, going back to the sequencing, I know there's been like a tremendous amount of effort put into sequencing SARS-CoV-2, but I know it's largely amplicon based. Is there any interest or effort in looking into co-infections or doing metagenomic sequencing of these patients?
Absolutely. So there's a number of academic labs that are doing that right now. We've got some activities at CDC where we are looking at co-infections and bringing a more metagenomic type approach to it. I think different labs have really employed different strategies throughout the pandemic. Many labs, when back at the start of last year, were using much more of a shotgun metagenomic type approach. But I think as time went on, as the cataloging and monitoring of variants became kind of the primary concern, many groups, just for cost effectiveness reasons, shifted over to amplicon-based strategies. That said, we've got a series of broad agency announcements that we've been putting out that have been funding research in and around this area, and some of those projects focus on co-infections. We're trying to drive that as much as we possibly can. But I think that is a key area of research, and I really have been looking at a lot of the literature on that.
And in terms of the emerging variants, I think I read that there is this theory that, for example, the P1 lineage emerged in immunocompromised patients in Brazil. Is there any targeted effort to sequence SARS-CoV-2 genomes from immunocompromised populations?
A few of our BAA projects are actually focused on that specifically. And that's one of the guiding theories around some of these long branch variants of concern that we've seen. I mean, it certainly has been floated around B117 as well, just because it doesn't have a lot of ancestral lineage that we can find, and it just sort of pops out mid-September with a whole host of mutations fairly stably fixed. I think it's certainly an area that I think will merit further investigation as time goes on. And I think both looking at immunocompromised as well as looking at these cases of frank vaccine failure are going to be two areas where I think people will specifically be looking for.
So if there isn't any more questions, then I think we'll just wrap up. And thank you, Duncan, for such a thorough and engaging presentation. And we will be providing a recording of this on YouTube so that you can send it to all your friends. And I will stay on for another few minutes in case anyone else has any questions. But other than that, thank you so much for your attention. And we hope to see you at some of our next respiratory pathogen series on bacterial, fungal, and other viral respiratory pathogens.